The paper that will be discussed here is Drosophila as an in vivo model for human neurodegenerative disease. The purpose of this paper is to discuss Drosophila as a model for researching diseases such as Alzheimer's, ALS, Parkinson's, polyglutamine diseases such as Huntington's, and through various methods of research, it discusses the fly model's important contributions to understand. So the first question to ask here is, why use flies? So flies are economic, they're cost effective, they're easy to regenerate, they do not require a lot of attention, they're simple to work with, and they actually model human genetics really well. The Drosophila nervous system is quite complex and has the ability to model neurodegenerative disease with outward symptoms. And sequencing and comparison of the fly and human genome reveals that there's a large amount of gene and genetic pathway conservation between flies and humans, including many genes that are known disease-causing genes for humans. So this makes it possible to also use a fly model to explore genetic pathways. This is particularly important because transgenic flies can be used to study new and old pathways and understand how certain proteins and molecular domains play a role in disease. Within the context of neurodegenerative disease, it's absolutely crucial to learn about how proteins interact with themselves and their surroundings to form the degenerate structures themselves. This figure is taken from a study that looks at HSP70 and its effect on the eye's degeneration from polyglutamine protein in Drosophila. HSP70 is a molecular chaperone protein from humans that is used during the folding process. So cell cultures in the past had shown a kind of a link between an increase in molecular chaperones and a decrease in polyglutamine inclusions. In this particular study, researchers took this information and they asked the question, does this pathway still affect organisms in vivo, and could increasing the use of molecular chaperones affect the level of neurotoxicity reflected? So to do this, they did a series of crosses. The control, outlined in green here, IA and ID, expresses HSP70 and no polyglutamine repeats. As you can see from the photo, the cross-section and also the close-up picture of the eye appears relatively normal and healthy. If you look at the next series of pictures, IB and IE, they express the expanded polyglutamine protein without HSP70. So as you can see from the cross-section and also the close-up image, this eye does not look very healthy and it caused degenerative structural effects. There's a lot of white area in the cross-section photo, which shows that there was neuronal death that occurred there. The last set of pictures, IC and IF, express both the expanded polyglutamine protein and HSP70. This last picture is probably the most interesting finding, that HSP70 helped reduce neuronal death. It mitigated the degenerative effects that you see from the polyglutamine protein in the photos from before. This shows that chaperone function contributes to the toxicity and progression of disease and neuronal death in Drosophila with polyglutamine proteins. The study also showed a link between the ubiquitin and proteasome pathway and the autophagy pathway, which is a pathway that helps to refold misfolded proteins in times of stress. And it's central to many processes, but is specifically associated with neuronal muscular degeneration, which is a major symptom for polyglutamine-associated disease. The link to this pathway could be why HSP70 was so helpful in reducing the degenerative effects. The fly model can also reveal the effects of proteins and molecules and pathway relationships, which was reflected in the results of this study. So how does this contribute to the paper? The purpose of the paper was to show that fly models are important to the scientific community and that they offer insight into human genetics. And the study did a pretty good job of that. Flies have shown that proteins and their function have a role in contributing to neurotoxicity, and earlier findings had only suggested that pathogenic polyglutamine tendencies were due to the protein. But now we're seeing from these models that it's in fact more complex and that there are proteins involved and chaperones and enzymes and all that. So as for the field, this can be applied to create other models, but it also leaves us with a question. Could understanding pathways help to mitigate disease after onset? More research would need to be conducted, but perhaps fly models could be at the forefront of it.